So I really want to take a second to thank the organizers, and especially Allison and Maureen for just making this a great conference so far. I'm really excited to be here. So um, I'm going to talk about this weakly electric glass knifefish a lot today. Eigenmani of erections. Now, for anybody who does control theory or studies stability of systems, I think this system is perfect because it has eigenvalues in its name. But it also has a number of features that make it really kind of an ideal model system for studying animal behavior, locomotion, and so forth. One is that it has good image forming eyes, as well as a second non-contact sensory system that is called the electrosensory system, where all over the surface of the skin are electroreceptors that detect a weak electric field that the fish emits with an organ in its tail. And so Malcolm McIver nicely describes this as sort of like touch at a distance. So objects in the nearby surround that have a different impedance than the local water distort the electric field, and that changes the transdermal potential, and the fish is able to detect that with electroreceptors embedded in its skin. And so it has two separate image forming modalities that allows us to do experimental manipulations to put those in conflict. But it also has this beautiful undulating fin that I'll refer to a lot in this talk. And it's actually an evolutionary adaptation of an anal fin. And so I like to tell engineers that, evolution works with an inherited plan, and sort of evolves what is an actually amazing adaptation that allows the animal to swim forward and backward nearly equally well without body bending. And then it can use body bending to move its electric organ sort of independently from its ability to locomote, which is kind of remarkable. But because it's an adaptation of an anal fin, it now has its anus here. And I don't know, I'm an engineer, I don't think that's optimal. Biology isn't necessarily optimal. I would probably disconnect the hose and put it back here on my underwater robot. But it is remarkable nonetheless. And here's a bottom view of that same fish facing the other way. These are its pectoral fins and its eyes. And if you study this fin for a while, you'll notice it has this beautiful sort of inward counter-propagating waves, these undulating fins from head to tail and tail to head. And they're driven waves, much like my fingers are driven. And so if the fish wants to swim faster in one direction, it can just recruit more of these fin rays one way than the other way and create a differential imbalance to the force, allowing it to swim nearly equally well forward and backward. I'm not going to talk a lot about that today, but I'll give a little hint into our studies into that. But the other thing, and actually I got introduced to this model system by who was my next door neighbor in Baltimore when I first moved there. He's a biologist. He was in a different building. I never would have met him, but I one time saw him, mowing his yard, and I introduced myself. And within a few minutes, he was telling me about the 4 parallel somatotopic maps of the electrosensory lateral line lobe, and his high pass filters and band pass filters and receptive fields. And I was like, who's this crazy guy sitting next to me, living next to me? So I went down to his lab, and he had an old fashioned what's called a pen plotter, but it's basically a thing that you can hook a voltage up to and change the voltage and move a gantry, basically like a really nice motor stage with a tube at the end. And he would move it back and forth. The first day I was in his lab, I'd never met him in his lab. We ended up collecting 4.5 hours of pilot data that was for our first successful grant. So it's just been a really exciting collaboration over many years, dating back almost 20 years. So that's Eric Fortune standing up in the second floor, moving his hand back and forth. And to me, it looks like this fish is attached to the tube with a spring and a damper, but the spring and damper are all in the fish's mind, much like maybe you could model the mouse following the trail, sort of getting pulled back to the trail, but it really looks quite mechanical. And so his arm was getting tired, so we used a motor to move the tube back and forth. And so you can see even very, very minute motions of this tube, the fish is tracking quite well. There's a little offset, there's a little drift, but it does really nice sensory motor tracking. Yu Yang, who's sitting here, who's now a postdoc here, who's a graduate student in my lab, studied the fact that these animals can almost perfectly phase match to that signal, and we think that's using an internal model principal controller. He just published that last year as one of the final second to last paper from the lab. One more coming.
So Noah,
Yeah,
I'm sure I asked you before in your previous talk, but is that visual information?
visual or electric, you can do it completely in the dark, and they'll track the tube as well, and just use the electrosensory information. They're much better at it when the lights are
It's a
on.
self-generated electrosensory bouncing.
It was, yeah, or maybe not bouncing isn't the right image, but it's sort of it's a electric field that's oscillating, that's sort of getting distorted by the objects, and then it can detect. It's not quite like similar to echolocation that it's emitting energy into the environment, but maybe the physics is a little different. And so they can do it based on electrosense, but they're quite good visually as well, and they actually track much better when the lights are on.
Yep. So it's clearly that they can do translational tracking. What about orientational tracking?
Haven't investigated, but I've anecdotally, when I go into the lab, I mean, a lot of fish have like an air bladder that would make them sort of flip upside down. So they stay upright. And when I look in the lab, when I look at the fish head on, you can see their fin kind of going like this. It's quite cool. And as I see the body of the fish go one way, I see the fish turn its fin the other way, looking like it's balancing, but I've never actually studied that. And Malcolm McIver's done a lot of really nice work of more omnidirectional motion, and another species of these called the black ghost knifefish that you can buy at pet stores because they're quite beautiful. They do quite, omnidirectional movement. George has studied with Malcolm the sort of heaving behavior, and I think the brown ghost. So they have the ribbon fin can enable lots of directions of movement. We do one-dimensional stuff in my lab. Okay, so what is this? This is now the underside of the fish, one snapshot in time, where there's a traveling wave from tail to head and a traveling wave from head to tail. And then what we're going to see over the time axis is let's track any one of these points, any peak or a trough over time, and it will drive toward the center and then extinguish when it meets the other traveling wave direction, right? And then the fact that this is oscillating back and forth is because while the fish is shown as stationary here, it's actually tracking a moving tube. And so what we're able to do is move the tube back and forth. The fish moves back and forth to track it. We then stabilize that here on this y-axis and do a fish relative point of view. And we can see the fin is actually oscillating this nodal position back and forth under active control. And if we do lots and lots and lots of trials, and this is unfortunately before ChatGPT, so a poor undergraduate clicked on, I think, 50,000 images. 50,000 clicks across, because multiple clicks per image. And you can see that we can sort of estimate a transfer function or a dynamical system between the movement of its nodal position and the movement of the fish. And this is the same reference, but multiple trials of the fish's movement, and we sort of estimate those dynamics. Yep.
This is learned, or this is
innate. Yeah, they just swim into the tubes, and you reach into the tube, move it.
No, I'm talking about juveniles compared to adults.
We haven't ever looked carefully, but we study fish from all different sizes, and they all do this really well. But we haven't ever looked sort of like developmentally. But part of the problem is you can't actually breed them in the lab. You just get them from aquatic fishery sources. Nobody's ever been able to breed them successfully in the lab. So it's hard to study them in that way. So I should say that an earlier study just looked at constant speed motion, and we sort of built a nice model that it turns out this movement of two inward counterpropagating waves gives rise to the difference of two sort of quadratic forces. If it's since it's a relatively high Reynolds number, I'm not a fluid mechanist, but this sort of was freshman physics, so I was able to sort it out. And as a result, you have two quadratic forces that are sort of mostly canceling out, and everything ends up looking linear to third order, which is kind of remarkable. I can walk through the math of that later if somebody's interested. It's not the main point of the talk, except to say that that's modeling study together with experiments that we did in Malcolm's lab, where we had a robot that we programmed to have counterpropagating waves and measured the forces. That was all published in our PNAS paper together, which is somewhere on here. And then this system ID study, where we moved it at different frequencies and sort of fit it empirically. We end up with a stunningly simple plant model, but where we actually can estimate, all of these terms, and then plus order third and higher. Now that's just the plant. That's the engineering parlance, for the thing that's being controlled by the nervous system. And the value of kind of, I don't know that we can really understand the nervous system if we don't understand kind of how the nervous system's output into the world gets filtered by dynamics and then returns back to the nervous system. And so this was sort of important for me as a control engineer to try to sort out.
What's the role of noise, and what is the role of estimation and filtering?
Let me come back to the estimation and filtering, and noise that's actually kind of what the active sensing is going to kind of get at. So that's a great question. So this is just, I mean, there's motor noise that I'm not modeling here directly, but that'll come up. This is just like the simplest lump parameter, undergraduate mechanical engineering model for what a fish swims like that we actually think is pretty well validated. But you're right, there's noise terms and other things that we're neglecting. One of the things that I think is remarkable, normally we would like to measure the muscle signals if we wanted to really understand what the output of the nervous system was. But the cool thing about the ribbon fin is it's oscillating at about 10 hertz. If you look at one point of the fin going up and down, it's about 10 hertz. So the nodal point gives us kind of an abstracted neural output from the spinal pattern generating circuits and so forth that's operating at a time scale above the time scale of actually 4F control. So I feel like it's like a good proxy for the direct motor output. Yep. The nodal point is this position in the middle. When if I go back to that one video, it's this sort of where the fin is sort of meeting in space. We call that we didn't have enough. I never came up with a better name. Or it's this sort of virtual position of where the two counterpropagating waves meet. Now this is really interesting for a lot of reasons. One is that if you do two sine waves, they don't meet at a point. They're sort of discontinuous, but the fin sort of smooths that out in interesting ways that we don't really understand. There's a lot of little details there that are quite interesting. Yep.
M is over there is the total mass.
It'd be the total mass
The
and added mass and the added mass. Yep.
And are there experiments you or Malcolm or somebody else has done or done where you just change the added mass suddenly, for example, by adding something to the body of the fish to ask how quickly or how slowly?
That's a good question. We haven't done that because it's hard to sort of add things to these fish that are quite fragile. To add a big mass to them or drop it off would be sort of tricky to do. That's it. People have done that in other animals. They've chopped off wings of flies while they're flying, or had masses drop off of the things, and that's an interesting. This isn't probably the best model system to do that in, but that's a good.
Or if you change the size of the tube, you effectively change the added mass.
That's interesting. Yeah, I have to think about the fluids of that. We did something, I'll say similar. Again, you did something similar but different, which is that instead of changing the plant dynamics, changed what the fish senses as a result of its movement. So he did a closed loop experiment where when the fish swims, he closed the dynamical system between the fish's motion and the tube's motion to change the effective plant dynamics that the fish has. So as it swims now, the sensory consequences of that swimming are different, and the fish does learn and adapt to that over about a 30-minute time scale. So, okay, so what's the picture point of that story is that, I'm a control engineer, so I like block diagrams, as we spent a lot of time trying to elaborate this block diagram over the years. Instead of just thinking about the nervous system controlling the nodal point, I like to think about there's this sort of like the nervous system is first setting up a simple dynamical system to control. That counterpropagating waves really simplifies control because now there's just one point that needs to be modulated, and you have really nice you get this damping term out of a second order quadratic drag model because there's these two inward counterpropagating waves. So by just simply setting up counterpropagating waves, you get a really simple, easy to control plant. Sort of using the kind of the open loop neural drive to simplify what the closed loop controller is going to need to have under control. So we figured this all out. Let's turn off the lights and go home. So here's another bottom view of the fish. It's hard to see because it's under infrared illumination. The lights are on right now. We're capturing in the infrared for a reason to become clear in a second. And then when you see the light bulb turn off on the left, turn a little icon to show we turned off the lights. And the fish behavior changes quite dramatically. Likewise, if we have the lights on, and then we'll turn them off in a second. We're looking at the bottom here. Yeah, it's gonna the light will turn on. There we go. And it just snaps into place, right? So in both of those cases, when the lights turn off, the fish starts doing this extra movement, and when the light turns on, that seems to turn off, not completely. I'll show you that later. This is just one anecdotal trial. The point is the fish is still tracking the tube, but it's also doing these ancillary movements that we have a whole suite of evidence that those are for the purpose of active sensing, as opposed to just like the lights are off, so let's party. And here's one really good piece of that evidence collected by Sarah Stamper. This is a pretty crude analysis, but what we did was we took the fish's movement with a stationary tube. This is a simplification, but I'll explain it this way. We take the fish's movement with a stationary tube under different conductivities. That's what the different colors are. When you increase the conductivity, you effectively decrease the electrosensory salience because it's like you're shorting out the electric field. And so when the lights are on, we see a very negligible impact of the conductivity, although it's notable that the fish's overall energy, I'm just plotting the Fourier magnitude of the fish's movement as a function of frequency. The overall energy of the fish's movement is higher in the highest conductivity, but maybe it's just noise. But when we do it in the dark and we increase the conductivity, the fish moves more and more and more as a function of this decrease in sensory salience. So that's our strongest that was our first strong evidence that this was an adaptive behavior for the purpose of gaining sensory information, because it was not affected just by turning off the lights, but affected by the electrosensory salience.
Can I ask a question?
Yeah, of course.
What sets that maximum?
What sets the, like where that peak is?
Yeah.
I don't have a great answer for that, but this is sort of a really crude analysis that neglects the fact that a Fourier transform sort of treats it as a stationary signal. We'll see there's actually kind of more of a switching that's going on that's a lot more interesting later. So let's come back to the question about those dynamics. Yep. Mostly they're ribbon fin. Their bodies are bending. They're doing all sorts of things that we frankly don't understand. We think a lot of that body bending is sensory. They'll put their head over by the side. When they move their tail, it's obviously that's where their electric organ is. So they're sort of like moving the flashlight around. It's extremely complicated and we don't really understand it. Tail beating does increase as well as a function of this. An experiment by my now postdoc. He was a rotation student in my lab while I was a graduate student, and then he wanted to come back to my lab for some reason as a postdoc when he finished his PhD. But just from his really nice laboratory rotation, he got this current biology paper, so it's quite impressive. So I was very happy to have him back. Where what he did was he said, let's take the fish, put it inside of a tube, and let's measure the fish in real time. And when the fish swims, say, forward, let's move the tube backwards, let's say 1 to 1. And if the fish is trying to regulate that active sensing in order to maintain a certain amount of sensory slip, then when you double the amount of reafferent feedback the fish gets, maybe the fish will move less. That was the hypothesis. And if you move the tube in the same direction as the fish in real time, then if the fish swims forward, it would have to swim forward more to get the same amount of sensory air. That's the idea in this experiment, is we varied this feedback, this artificial feedback parameter gamma, and see how that looks. So first, a little tiny bit of math. After a preliminary set of experiments, we realized that correlations were going to be a funny thing with respect to gamma, but we expect that when gamma reaches a gain of 1, that's where the fish can't move inside the tube, because as the fish swims, the tube moves right along with it. That should be some kind of an infinity. And then if we want to make the fish receive the maximal sensory feedback, that's when there's a gain of negative infinity. So if the fish goes, say, 1 centimeter forward, the tube would go infinitely backwards. So through this hyperbolic change of variables, we get this prediction that the fish's RMS swimming ought to be this scalar beta times a sort of desired average. So this is what we're going to regress on. What's this beta relative to the error signal? So I'll explain that again in a second. So let's just think intuitively for a minute. If we move the fish in the opposite direction of the feedback, that enhanced the feedback, and we see that the fish's motion was sort of small. When we moved the fish in the same direction, but about half of the amount of fish, the fish's movement was larger. So it did seem that the fish, and I'll just show the next slide, that is a function of either the augmented original gain or this hyperbolically transformed gain, that the fish increases the RMS of its movement as a function of this feedback gain. And maybe more importantly, if you look at it in error coordinates, the fish maintain nearly constant sensory error. I should say it's the RMS sensory error. How much error feedback the fish is generating is constant over a wide range of these gains. tailing off a little bit at the highest gains, because that's when the fish has to move the most, and maybe then it's an energy information trade-off. But roughly speaking, the fish maintains kind of a robust level of sensory error all the time. So it's sort of dithering enough all the time.
And the fluctuations themselves are also a function of how close you are, gamma -1 equal to when gamma is close to 1.
When gamma is close to 1, that's the same as beta being large, and that's when we see eventually the animal sort of gives up a little bit. Exactly.
And as it gives up, the fluctuations starting to essentially diverge.
The actual fluctuations are diverging of the fish, but the amount that they're diverging is becoming sublinear.
Can you quantify how quickly those fluctuations are diverging as gamma approaches 1?
Well, I mean, I think it was individual experiments over a certain epoch at different gains, and sort of this curve kind of quantifies that.
I was looking at them.
Okay. The other thing that could be happening, and this is where we almost like sent this in before thinking this through. This is one of those like gotcha moments where like, oh my gosh, maybe what happened was we changed the feedback of a dynamical system, and the fish was doing the exact same thing every time, but now we've changed the closed loop dynamics, and all of what we're seeing is just an epiphenomenon of us changing the feedback. If that were the case, then well, it turned out not to be the case. So if we assume a static control system, then what we can do is infer how much kind of active sensing is going on by basically taking the fish's RMS movement, kind of undoing the transfer function dynamics and figuring out what the power spectrum of this A of t would be. And it does indeed change as a function of the feedback gain. So this is just a computational control to say, we think the fish is actually sort of intentionally changing how much it's moving in order to regulate this sensory slip. And the fact that it changed how much it moved was not an epiphenomenon of our change in the closed loop dynamics, just kind of a subtlety. So what's the summary here? Active sensing movements increase as a function of sensory stimulus saliency. I should say active sensing movements increase as a function of decreased stimulus saliency. So when we increase the conductivity, the fish swam less. The way I like to think about it is the fish is now reaching out through its locomotor dynamics in order to modulate sensory feedback to enhance sensation. But I want to take a second to pause. So first of all, there's a lot of definitions of active sensing. One of the most common is that the electrofish generates an electric signal, right? Emits it into the environment. Just like a bat generates an echolocation signal, emits that into the environment. That's a common notion of active sensing. In engineering, active vision is often where you take lights and shine them in a structured light and shine them on an object and move that structured light around. That's an example of emitting energy into the environment. The most common type of active sensing that we all do is active sensing where we generate movement in order to enhance sensation. So if I want to measure the weight of this cell phone, I'll put it in my hand and I'll sort of make sure I excite the deeper cutaneous receptors and the proprioceptors and so forth, the stretch receptors in my muscles and skin and so forth to try to estimate its weight. But if I want to measure its texture, I might rub my fingers lightly over the surface. Those are examples of movement-based behaviors that enhance information. But I've increasingly become of the opinion that most of these not most, that's an overstatement. There's an interesting and large sort of subset of active sensing behaviors that don't need to be thought of as an information-driven phenomenon. And I'm going to motivate that next, which is to say, if I have sensors that have a certain property, then I can just set a control objective, and active sensing becomes necessary in order to achieve that control objective. So I'm talking about this as if the animal is doing something in order to sense, and in a sort of a subsystem way it is, but I don't need to, if I want to make an optimal control model or even kind of a simplest model, which I'll show, I don't need to bake the need for sensing into the model. I can just say, I want to control myself to stay still. Let me do the best job I can of staying still with the least amount of energy. And it'll turn out I'll need to do some active sensing to do that. And I didn't have to let me also add an information term to my optimization criterion. I just had to say, I want to stay still. Okay, that was the closed loop experiment that Devajoti did. So let me get back to the question of why do active sense? So first is this idea of perceptual fading. I want everybody to kind of sit as still as you can. And it might be hard for the people in the back. If so, look up something called Troxler fading. And what I want you to do is fixate on this plus sign with keeping your head and eyes as still as possible. And especially the people in the front row should just see the purple dot slowly fade away. Okay, it's a really powerful illusion. It's quite effective. Our eyes, things disappear before our very eyes if our eyes don't move relative to the scene, especially in the visual periphery, but even in the fovea, it's just harder to do for the fovea.
That's the only thing we're supposed to see.
Did you see it or not?
I'm seeing a green circle.
No, but if you fixate here without darting your eyes around.
There is a green circle.
What?
There is not.
There is a green circle moving around. There is no, actually, I think it's not that there's a green circle. I think that the purple is disappearing. Fixate on the plus sign. And you actually don't need this sort of thing moving around that works without it, but it works a little better with it there for reasons I don't understand scientifically. But if you fixate on that plus sign, the periphery largely disappears. It's very hard. You can't voluntarily suppress your eye movements, but this suppresses them just enough that the peripheral vision disappears. So what's happening, and people have known this for a long time, there's been huge debate in the oculomotor literature for the last like 40 years about what the role of microsaccades and drift and other little tiny eye movements are. But one of the prevailing theories is that they're actually for the purpose of refreshing the sensory retina, because otherwise you get perceptual fading. Chris Neal's lab did beautiful experiments in a freely moving mouse where he was able to reconstruct, his lab was able to reconstruct the eye movements of the mouse while it was doing a prey capture behavior. And right at the moment that the animal does eye saccades, you see these large increases in responses to visual neurons. So it's not only do we have this kind of cognitive perceptual fading, but even in the early visual system, the visual system is very sensitive to change, but not very sensitive to absolutes. Well, engineers inspired by this idea of adaptive sensing that's not, okay, so biological sensors are not very good to absolutes. It's, why you stop feeling your shirt while you're sitting in a toilet. We only measure change. You can move around and see, okay, good, let's just throw my shirt on. We can sense when there's movement, but we can't sense when there's a constant. A really fun illusion to do is to put your hand in two different baths of water at different temperatures for a few minutes, and then put them both into a central bath at the same temperature. And the hand that was in the hot water will feel cold, and the hand that was in the cold water will feel hot, even though they're in the exact same temperature water, right? So we're really good at change detection, but not very good at absolute. Engineers don't build sensors that way. We spend thousands and thousands, the sensors that go on the underwater robots that my colleague uses are like 50 grand, and they're designed to be, perfectly accurate with very low drift. It's extremely hard to do. No animal has a sensor that's that absolute and precise, right? It's just they're all good at measuring change, and they're not good at measuring things perfectly. I'm making very gross, overgeneralized statements on purpose. Please beat me up about it. So engineers built one of these adaptive sensors called an event camera, this is what the event camera gives you. It gives you nothing when there's nothing moving, but when something's moving and there's a change in a pixel value, you get an event, either positive or negative event. And so those are sort of a biologically inspired sensor, and they become very popular in the computer vision engineering literature because they're extremely low latency and they're incredibly high dynamic range, because every pixel locally adapts to the current light level. So, if you take a photograph with a really good camera when you're driving through a tunnel, either the tunnel is too dark, or if you get the tunnel light enough, then the end of the tunnel looks completely oversaturated and blooming, right? You sort of can't get both. You can't get your aperture set and your exposure time and so forth to capture a high dynamic range. But if every pixel is locally adapting to whatever the brightness background is, then you can get a super high dynamic range. And then you also only have to have this camera send information when there's a change in the image. So that was the idea. So biological sensors are adapting, as I mentioned, and they're kind of event-like. They're not really event sensors. I don't think of them that way. I think of them more like as little like local high pass filters. Because they're good for detecting change and not absolutes. To your point earlier, event-based cameras create real problems for state estimation. Because if you can't measure absolutes, then it's very hard to sort of infer your state. So I'll see if I can motivate this a little bit. So let's say we're an electric fish swimming inside of a tube, and let's model the tube as giving a kind of voltage pattern that looks like this, or a light pattern, whichever you prefer. And then what we're going to do is we're going to stick, for simplicity, let's put one sensor on the side of the fish, and that sensor is going to pick up either like the 0, and then a 0.5, and then a 1, and then 0. So we're going to get an S of X, where X is the position of the fish, and S of X is the sensory measurement that the fish gets as a function of its position. So that's nonlinear. And then I'm going to use, for convenience, I'm going to use Z as the fish's velocity. And so now I can take that MX double dot model that I put before and make a simplified normalized parameter version of it, where I say position dot equals velocity and velocity dot equals minus velocity. So that's the dampened part, right? Then what I'm going to do is I'm going to make what I'll call a minimal perceptually fading sensor. I'm just going to take the time derivative of my sensor measurement. So now if I put my little sensor in front of that black and white bar grating and I stop moving, I'm going to get 0. But if I start moving by an edge, I'm going to pick up a positive number. If I start, just like the event camera, right? Except rather than events, I'm going to get an analog signal out of it. And I did leave off a term here, I'm surprised, plus U, the control input. The control input, think of that as the nodal point, okay? So I'm going to write this down. And we'll refer back to this a lot. And then the output I call Y equals, and we'll just use the chain rule, that's gamma of X. So that's a very simple coefficients.
Yeah.
Okay. Unity coefficients. Yeah. When we do the actual model in the animal, we incorporate them, but it's not critical. So now if we linearize these dynamics around a desired equilibrium, let's say the fish wants to sit by one of the windows, and we linearize the dynamics. Well, the dynamics are already linear, but this output is nonlinear in an important way. And I should say, if we didn't take the time derivative, we wouldn't get this lack of observability. But lack of observability means that for the linearized dynamics, I cannot infer the fish's position in a neighborhood of that equilibrium. So I can walk through that if somebody's interested, if I have time at the end. But linearizing this basically means I can't figure out the fish's position. Now, there's a couple of ingredients that are necessary for that to be true. One is I can't have a spring term in the model. As soon as I break symmetry in position, then the position becomes observable. But for a translationally invariant system like a fish swimming in a tube, which one can argue whether or not the fluid dynamics are the same in every spot in the tube, but let's keep that thought. Then you wind up with this lack of observability. Gamma is just S prime. It's just by the chain rule. I took BDT of S of X, which is S prime of X, times X dot, which is Z. That is
the assumption that it's a function only of X and not X dot.
correct, because what it is remember it's the voltage on the surface of the skin as a function of the animal's movement. Yep. So think about if you gave a little like a middle school sort of robot project, you put a little photo detector over, say, a stripe on the ground, and what I'm doing is that photo detector will be giving me a value that was a function of the robot's position, and that's what S of X is. But now I'm going to make it hard by taking the time derivative of that, so I only know when it's changing. As our simple model, the way adaptive sensing works. And so Eduardo Sontag saw this result and came up with a nice proof that showed that even though the system is linearly unobservable, we had this theory from Kuhner-Peretti that we at least had local nonlinear observability, and I'll explain what that means in a second. But first I'll start with Eduardo's result, which says that the system is globally observable if the gamma function is aperiodic. Otherwise, it's only local. And that makes sense because I wouldn't necessarily know where I was, which window I was next to. If it was truly a periodic sine wave signal that was my scene, I wouldn't be able to tell where I was, whether I was next to the first window or the second window. But the idea of observability is as follows. If two states are different, two X's and Z's are different, then there exists a U that when I play that U, eventually my Y's will be different. My outputs will be different. There exists an input that will make any two states discernible. And that, and then there's other theories that says if I provide a sufficiently persistently exciting U, then I can tell apart all states and so forth. But the point is that control engineers use this linear notion of observability, which doesn't have anything to do with the input. Even though in reality, what this is saying is I have to provide the right kind of input to make my state observable, which is a complete paradigm shift from basically every control engineer who builds a guided missile or who makes an underwater robot or whatever. They all assume separability. They say, I have sensory measurements. I'm going to build my state estimate, and I'm going to apply that state estimate as if it's the God-given truth. And I'm going to do my control and my path planning and everything else based on that. This is saying, no, hold the phone. What I need to do is make the appropriate movements that make my system observable so that I can figure out what my state is, and then I can actually try to control it. The flip side is a very simple, there's a very simple proof that if this is my dynamics, I just copy that and put it down here, remember my U this time. If I put those in as my system that I'm trying to control, I was able to show, actually, Eduardo didn't believe me, and we were at a poster session, so I went off and wrote the one-line proof. It's very simple. There's no way to build any dynamic feedback that stabilizes an equilibrium point. It's impossible to stabilize an equilibrium point for this system. So it's non-linearly observable, but it requires you to be moving in order to do the observation, and you can't stabilize an equilibrium point. So try as it might, the fish can't stay still in the tube, even if that's its goal. Now, I'm not saying that's its goal. Maybe it has other reasons it wants to move around, but it's impossible to stay still if you have adaptive sensors and you want to stay still, which is kind of unfortunate. But you can do the next best thing. You can build in a small oscillation, and then you can prove that you can actually get stabilization of a limit cycle. And so that was a paper that just came out last year, and we're working on a journal version of that. I mean, we have a journal version, I guess, because this was a joint issue, but then working on a few more extensions to that theory. So the idea is actually very simple. You just add a little active sensing oscillatory component, sort of wiggle back and forth, not like the fish. In this case, it's just a pure sine wave. And then you can apply a feedback term, and you can asymptotically stabilize a desired limit cycle. Where this is now the XZ plane, and I can sort of stabilize a small oscillation. And you can think about the trade-off in active sensing between sort of active sensing, which is like an explorer behavior, and feedback, which is this gain K. It turns out they have to satisfy a certain rule in order to get stability.
Just so that I don't get lost, sorry, this is only adapting sensors, that's the point, right?
Adapting sensors, yeah. Yeah, so it's a limited case.
Yes.
But I will say one thing. Any sensor with even like partially adapting or noisy, that even if it doesn't adapt perfectly, then when you add a statistical desire to stay close, there's still a motivation to do active sensing. Yep. So it generalizes a bit to the non-perfectly adapting case, and to when there's a symmetry breaking term, you can do numerical exercises to say, in those cases, you still also emerge with that active sensing as well. Yep.
Your model for the visual are they not adapting as much?
So it turns out they oscillate in the light as well. Yep. So I'll come back to that. Show some video talk things about that. Yep.
Possibly very naive question. Like in computer vision, you take two images from different locations, and you can do that because also people do super resolution from it.
Do what regulation?
Super resolution.
Oh yeah.
You you think there's also super resolution that is useful, or do you think it's mainly this?
That's a good question.
Resources?
Yeah, I don't know. That's a good question. Those are kind of questions for people who look at like higher order visual reasoning, maybe primate studies and things like that might address that kind of thing. I don't know if the fish are doing anything like that. There's nothing back here.
Sorry if I missed it. Do they also have basic mechanical like shearing or touch senses?
They do it. They have mechanosensory lateral line senses. Most of the time they stay off the wall, so they're not directly touching, although they do sometimes seem to sort of nose tap the sides.
Can I ask about how much sensing or how much ability you have as a function of the properties around? Maybe it will come to that.
A little bit. So it's in our paper. One of the things that's, so what we find is that the amplitude of alpha is term A. Now, this is obviously also frequency dependent, and I'm ignoring that. If you increase frequency, that's another parameter. This is all non-dimensionalized. You need A squared times K ends up being a parameter that you can do that ends up being the sort of critical parameter for stability for the system, and it has to be below some critical value in order to get stability. And I don't think I have any of those results here, but
And you've chosen the particular value of the phase.
the phase.
You've chosen A times cosine.
Oh, yeah, so I just, you can put an arbitrary phase here and it would still converge to that.
I
So
understand that, but is there something interesting about the dynamics of the phase? It's typically a slow variable, and therefore you can essentially often sense a lot by measuring or looking at phase.
that's interesting. I don't know. I'll give you an answer to a slightly different question, which is that stability based into this is quite phase dependent, meaning that this is not a globally stable system, at least numerically. We can prove it's locally stable, and numerically we can show it's not globally stable, and it really depends on sort of the phase you are. Like if you start at this initial condition at a different time, it might diverge. So that's a different question. But that's the one I'm gonna answer the question I know the answer to rather than the one you asked.
We can talk.
Okay. So okay, so engineers, as I said, almost invariably use this separation principle, which is a principle derived for linear time invariant systems, which says the best thing to do is actually to build an optimal state estimator called a Cowan filter, and then apply that state estimate as if it's the God's given truth. They take that linear framework and they apply it to every nonlinear system, and it works pretty well a lot of the time. Why? Maybe it's because we use $50,000 sensors, LiDARs that are perfect, things like that. And maybe if we built sort of more sensors that were a little sloppier but had super high dynamic range, we would no longer be able to do this. That's my contention. I don't know why I'm bringing that up again. However, the sensor is very different. Yeah, the biological sensors are really different. I don't know why I put all these things up here. I just said all that. Okay, so maybe in case I forgot. So the nice study that Devajochi did, which will pretty much wrap up my talk, is this more detailed temporal analysis of the fish's movement in the tube, and it's not just sort of Gaussian noise. It's kind of more interesting than that. So let's go back to a case where the lights were on. I showed a little short window where it was tracking a sine wave and it didn't look like there were any active sensing movements. But if you wait long enough, you see these larger bouts of movement. And if we take the time derivative of those, we see the fish is sort of mostly station keeping, maybe with a little drift that's you can see in the position, and then it does these larger oscillations and then goes back to station keeping. And if you take a kind of simplified, or I will say a nominal explanation for this, we can call this exploit, meaning you're kind of doing the engineering thing of just taking your best estimate and applying it for control, interspersed with these exploratory movements. And those are kind of like our working definition of explore and exploit. And if you do the same thing in the dark isn't just randomly moving all the time. You still see these fairly long flat spots of very little movement, but now we see much more frequent exploratory movement. So it's quite sort of a nonlinear switching type behavior where it's not just persistently exciting all the time, like in our simple sinusoidal model.
Is there something interesting about the statistics?
Yes. So if we look at a lot of those trials, now what I'm going to do is I'm going to take multiple trials from one animal doing station keeping in the dark and in the light, and I'm going to do a histogram of the velocities, and we see that we see this kind of what some people call a high center distribution, or this sort of broad shoulders and sharp peak in both cases. And if we try to fit that with and I'm plotting this on a log scale. So if it were Gaussian, it would look quadratic, because log of e to the x squared is, minus x squared or whatever. So the pink here is the best fit Gaussian, which is not very good. And I don't really care about the sort of order, but the blue curve is the fit, I think, of 3 Gaussians. But I don't care how many. It's just the point is that it captures this kind of high centered broad shoulder distribution. The kurtosis, which is kind of can be used as a measure of normality, when it's around 3, that's a normal distribution, and it's higher in the dark and much higher in the light. This actually surprised me because I thought, oh, when it doesn't need to do active sensing, it's going to look more Gaussian because it's more linear. But it's actually the opposite. When it only needs to sense infrequently, you get this exploitative behavior with just occasional exploratory behaviors. Yes.
When I reuse the data on the distribution, because it looks asymmetrical in a way, like light and dark is not in the same direction.
Good, yeah. So the fish is asymmetric, and we're sort of treating it like a spherical cow. I mean, we're sort of maybe it prefers to dart one way a little faster than the other, and things like that. So some of that's probably going on. We haven't delved deeply into that. Initially, we thought this might look more like, levy flights or some kind of a broad, we were thinking we'd see a heavy tail distribution. We saw no evidence for that, but we saw, so instead we call it a broad shoulder.
Is there anything interesting about the ink? Is the velocity corresponding to about a centimeter a second? Or
Right here, yeah, so that is, we use that as a heuristic for identifying when the animal switches between explore and exploit. So it's a heuristic. We've also done a lot of models where we can just directly look at the time domain data. Whenever the velocity exceeds a certain threshold, we say, aha, now it's in the explore mode. But actually, as the velocity slows down and reverses, it might still be in the explore mode. So we can make models where there's a hidden state and try to sort of do model fitting, but it all leads to the same idea, which is that we can have kind of a Markov process model where we have the animal's in an explore mode, and then it switches to an exploit mode and back and forth. And so, we mentioned turning on and off the lights, but Malcolm McIver's group had some data from a paper where they only had electrosense and they jammed the fish. And I'll just say when we reanalyzed their data, we had jamming on and jamming off. And we saw the same trend where when the jamming was off, they had a higher peak. Similar to when we had just the dark, but when the jamming was on, it's practically a Gaussian. Now their data were a little different. We had to filter it differently. So the statistical comparison is not exactly the same because we were working with somebody else's data, but we saw the same trend. So that seems to hold across modalities. When you increase the sensory salience, you go to sort of broader shoulders and sharper peak rather than just sort of all more like a persistently exciting strategy when you can't see very well. So what about other organisms? Well, Devajyoti was a hero, and he went around to a whole bunch of different papers, emailed lots of labs, and we found no counterexamples to this. We found some data that we just couldn't get our heads around, but everyone where he had like good station keeping data, he saw the same result. And this is for lots of different sensor modalities and an extraordinarily wide range of taxa. And in every case, we found sort of broad shoulders and sharp peaks. And eyes are like spectacular about this, and that was kind of well known from all the studies on eye movements. So we think there's kind of a basic fundamental behavioral design principle here because so many different organisms from single cell organisms to animals across taxa kind of have a similar strategy. And I know I'm out of time, so I'll end on this. So this is the point, I think, of my whole talk, which is that active sensing is a control problem. So if you formulate an optimal controller, well, first of all, I formalized a minimal controller that couldn't do station keeping and required active sensing. But if you take kind of a normative model framework and say, let me make an active controller where I just want to minimize my position and my effort. Out of that, it's hard to actually solve it mathematically, but you can build a very simple controller where you have a state estimator and a mode switching controller that occasionally turns on an active sensing term. And when does it turn on that active sensing term? You just keep track, it's a heuristic, you just keep track of your central covariance matrix of your state estimate, and when it gets large enough and you're sufficiently uncertain about where you are, you turn on active sensing, and when that gets small enough, you turn it off. And when you do that, you recover the broad shoulder distribution, you beat persistent excitation, and you do so using less energy. So we think this mode switching, we can't prove this yet, but it's better than the best sort of small persistent exciting strategy, both in terms of energetics and average state error. And so I'll just leave this up and I do want to show one last thing. Sorry, I got to show one last thing. So here's the event cameras again. Self-location is not reported when you're not moving because the event cameras have this perceptual fading. These guys at Northeastern right here in town build a system that uses an event camera to position a robot in front of a Gaussian scene using our control model. And here, I'll just let this video show. It's showing. It's going. Yeah. So we're doing this sort of oscillatory strategy, not the mode switching strategy. And all at once, the scene shifted and the robot now oscillates around this positions kind of off to the right here. And then in a couple of seconds, you'll see the image shift a little bit to the left, I think. Negative is to the left. And it sort of, the robot doesn't know that, but it then oscillates around that new position. If you try to just control this robot in front of the thing with a event camera, it would just sit there. It wouldn't know where it was. It has to get moving, and then suddenly it knows where it is and it can control it. So didn't want to talk about that video. Okay, I'm done.
Thank you so much.